Hello, I'm Christos Davazikos. I'm with the Center for Biomedical Image Computing and Analytics at the University of Pennsylvania. It's a great pleasure to be part of this session, even though remotely. And I'll be describing some of our work on machine learning and big data analytics in brain aging, MCI, and AD. It reflects work of a lot of people, but two of them are particularly have contributed to this, and their co-authors, Loanna Skampardoni, who is with NTUA in Greece, and Mohammed Habes, who now is with the University of Texas, San Antonio. So machine learning has been with us in neuroimaging for almost 20 years, and I was kind of lucky to be one of the first people who started to play with these tools back in 2002, doing mundane things at the time, looking at differences between men and women. Later on in 2005, we did something more meaningful, tried to find an imaging signature of schizophrenia. And since then, of course, there have been a lot of people, hundreds of publications that have looked at different methods and different applications. Machine learning is a little bit of a black box, trying to find the right combination of features from different parts of the brain and different modalities, different types of data, and comes up with a signature that is positive if you have with a biomarker that is positive if you have the signature of the disease, for example, and negative if you don't. We've been working particularly with Alzheimer's and aging, as well as schizophrenia. We call these scores the SPARE scores, spatial patterns for recognition of something. Back in 2007, one of the first successful examples we had in AD was this one, where we showed that there's an imaging signature of Alzheimer's disease that almost perfectly separates patients from controls on an individual patient basis. But of course, the goal of classifying, the goal is not really to classify people who are obviously demented here using expensive MRI and sophisticated tools. But the goal would be to identify these imaging signatures at earlier stages, such as MCI or even cognitively normal states. And this is some work we published back in 2014, where we showed that a combination of the SPARE score, the imaging signature, the structural imaging signature of Alzheimer's disease, ADAS-Cog, a cognitive test, and ApoE4 genotype in combination were very good at predicting which of these MCI individuals would progress to Alzheimer's disease over several years and who wouldn't. And of course, the holy grail would be to show it that cognitively normal people. And that's something we had, the publication we had in Brain with data from the Baltimore Longitudinal Study of Aging, where we found that actually, to our surprise almost, that the rate of change of SPARE-AD, so the rate of progression of this imaging signature of Alzheimer's, was predictive of which of these cognitively normal people would progress from a normal to MCI within an 8-year period. And this, of course, gives us a window of opportunity of 10 or 15 years before people become demented, or even more, to be able to intervene. So enough of the old stuff. I'll talk a little bit about the current stuff we've been working in, or the more recent stuff we've been working in the lab. And we've been trying to address two main challenges that I see in the field. One is reproducibility and overtraining. There's been a lot of hype about machine learning lately, and there's been a lot of studies that showed very good classification in a setting or another, but they don't generalize well. And the reason why they don't is because they have overfitted into a particular data set certain characteristics of a particular population. The second issue is heterogeneity. Oftentimes we're trying to find an imaging signature of disease, but the disease is heterogeneous. It has multiple signatures, it has multiple subtypes. And we've started to see that both in MCI and AD, in schizophrenia, in depression. And so therefore, it doesn't make sense to be talking about a single disease signature, but we need to come up with tools that find multiple imaging signatures within a population. So we have formed one, we've been involved in several consortia. One of them is the iStaging Consortium, which is now in its 3rd or 4th year. It's an imaging-based system for aging and neurodegenerative diseases. And we've pulled together data from over 15 studies. It's now 56,000 unique subjects. Many of them have longitudinal scans. More than 6,000 have longitudinal scans, so on average, three scans. Many of them have amyloid status and so forth. UK Biobank has been a major contributor, of course, here with all the functional data and structural data. And the first problem we solved and published a paper recently in Neuroimage. This is Ray Pomponia, who was the first author from my lab, and Hao-Chang Xu, who is a major collaborator from biostatistics. We used the combination of combat and a spline-based generalized additive model to capture nonlinear effects of aging. And combat is a method that has been used in computational genomics quite a bit to harmonize data from different batches. And to our pleasant surprise, we saw that we can actually put together data from all these disparate 15 or 16 different studies. Most of them are multi-site studies to begin with. So you're talking about dozens and dozens of different scanners and protocols. And we can properly harmonize them using comparable sets of controls. You can come up with brain aging curves of different brain regions like the ones you see over here. We've applied similar methods to resting state fMRI data. So now that we put all this together, we can start playing with this complex data and look at the brain charts, for example, not different from the charts that meteorologists and other scientists are looking at. This is Mohammed Habis, who used to be with us. Now he has his own lab at the UT San Antonio. And we've been playing with his brain charts, and you start seeing very interesting things. For example, if you look at if you develop a machine learning based index of brain aging, the SPARE BA score as we call it, and you plot it in color code here against the age and white matter lesions in the brain, you see that, for example, a 61-year-old who has a lot of these lesions looks 10 years older. His brain or her brain looks 10 years older than the brain of a person at the same age who has few of these white matter lesions. Similar results can be seen here if you follow this isocontour, if you essentially slide along this isocontour, which is not different from these isocontours here in these temperature maps. But what you see here is that the 62-year-old and a 74-year-old have the same memory performance, but of course they differ in white matter lesion load. So these white matter hyperintensities make you 10 plus years older, so they're not a good thing. Here's a plot that shows the evolution, the progression of SPARE over age. As you see here, it goes up non-linearly, which is not good news, of course, but unfortunately that's what it is. And it shows that this spatial pattern, the AD-like spatial patterns of atrophy evolve, especially after the age of 70, they start to develop rapidly. One of the most interesting and provocative findings has been that white matter hyperintensities double the probability of testing amyloid positive, which is very interesting, of course, because oftentimes these two are considered as different pathways towards dementia, oftentimes synergistically contributing. But here we see that there's an association between them. Of course, like everybody else, we've been playing not only with conventional machine learning methods, but also with deep learning methods. Several people in my lab, and this is Vishnu Bashyam, who's a new graduate student who has developed this monstrous deep learning network for estimating brain age. This was a paper we had in Brain recently. It was tested using cross-validation in a number of these studies, and also tested independently on the UK Biobank, or here the SHIP study from Germany rather, and it gave pretty good results. We learned a couple of things from this. The one thing that we learned is it's not a very good thing to be overfitting the brain age models. So you can get a very good estimate of brain age, but it might not be informative about the pathologies. The second thing that we learned is that if you want to specifically focus on diseases, and we tried Alzheimer's, MCI, schizophrenia, and depression, you are better off starting with this brain network here that has been pre-trained on this large number of MRIs, even though it looked at brain age, rather than trying to train a network from scratch. The second challenge that I mentioned in the beginning is heterogeneity, and we've been working with this for several years now. This has been work by Erdem Varol, who used to be my graduate student, and he's now postdocing at Columbia University. He is looking for faculty jobs. He's highly recommended, very smart guy. And Aris Sotiras used to be with my group, and now he has his own lab at Washington University, St. Louis. So we developed this method that instead of looking at a single imaging signature of disease and recognizing that there might be subtypes of disease, we found a convex collie top we find convex polygons, which basically are multiple imaging signatures that potentially capture multiple disease subtypes. So we've applied it now to a few studies. And one of them was in heterogeneity in MCI and AD, where we actually, even in ADNI, which is a fairly homogeneous set of subjects compared to the general population of people, of older people, especially people with MCI and AD. But even in this population, we found that there are 4 subtypes in MCI and AD. 44% of them had full-blown AD type of atrophy. 15% had this very focal hippocampal and medial temporal lobe atrophy. 28% had a very diffuse atrophy that involved the hippocampus. That's why they were enrolled into the study, but it was very diffuse. And then 13% had basically normal structure. And these subtypes had different survival curves. There were some interesting differences in many of the biomarkers that I don't have time to go through, but it shows you that even in a group that we expect to see relatively more homogeneous results, we have subtypes that are identified by this method. We've been applying this method to brain aging data from eye staging. And I don't want to steal the thunder of Loanna and Monica, who have an oral presentation in this conference. So I would urge you to go and look at their presentation, which describes our first attempt to dissect the heterogeneity of brain aging and find patterns of deviation from typical brain aging. So I would like to close here with just mentioning that machine learning is a powerful tool. It requires a lot of data to be useful and reproducible and lead to reproducible models. And even though typically people think of machine learning as a black box that gives you the right answer for, say, classifying disease or predicting progression, I also find it useful in a different way, which I think has become very important, and it leads towards dimensional neuroimaging. It allows us to condense this very complex data of millions of variables down to maybe a few dozens of variables of dimensions. Perhaps some of them are disease subtypes, some others are structural covariance or functional covariance networks, amyloid deposition patterns, and so forth. And it helps us distill this very complex data down to a small number of dimensions to better understand the heterogeneity of diseases. So I would like to end here by thanking a number of people who have collaborated with us in eye staging, and let some room for questions. Thank you so much.
Welcome, everybody. I want to present you data about decoding the neurodegenerative mind with pattern recognition in MRI in combination with meta-analysis. So firstly, I want to introduce the clinical features of frontotemporal lobar degeneration. Second, I want to discuss the imaging features that we have extracted with meta-analysis. Third, I want to show you how we can individually predict diagnosis and differential diagnosis in these kinds of disease. And in the end, I want to discuss the perspectives for application in clinical routine. Let's start with the clinical features of frontotemporal lobar degeneration. Frontotemporal lobar degeneration, or FTLD, is an umbrella term, and it includes a lot of diseases. So first, behavioral variant frontotemporal dementia, a disease affecting mainly behavior and personality. And on the other hand, we have so-called primary progressive aphasias affecting mainly language functions, 3 different subtypes, one fluent variant, that's so-called symmetric variant PPA, and on the other hand, 2 non-fluent variants. In particular, this is non-fluent achromatic variant PPA and logopenic variant PPA. All these different subtypes of frontotemporal lobar degeneration are characterized by clinical features. These clinical features define the disease and lead to a possible disease. Whereas we can also add imaging biomarkers, and these increase the validity of the classification, increasing it to a probable disease state. In my talk, I want to show you how we combined meta-analysis to identify disease-specific networks, imaging studies investigating patients based on multicentric data, and pattern classification to predict diagnosis and treatment in the disease. Let's start with the imaging features and results for the meta-analysis. In recent years, a lot of studies have been published on MRI and dementia. So we can conduct systematic and quantitative meta-analysis based on these studies published in PubMed and use these data to do big data mining with meta-analytical tools like Ginger Ale and SDM or seed-based demapping. Here are the results for frontotemporal dementia, the behavioral subtype. You see spots or maxima in the frontomedian cortex and in the anterior insula. We have redone this analysis by separating different imaging methods. Here, glucose metabolism, FDG PET, shown in blue, and MRI, structural MRI, illustrating atrophy in red. And again, you can see that frontomedian, anterior insula, and the parts of the basal ganglia are affected, and we see a separation or dissociation between the different imaging modalities. If we go to other meta-analysis involving healthy subjects, as shown in the right panels, we can see that there is an overlap. Specifically in the frontomedian cortex, we see an overlap with the neurocorrelates for social functions, self-related functions, and anticipatory functions, as investigated in imaging, functional imaging studies in healthy controls. We have also done these meta-analyses for the different PPA subtypes, as illustrated in the panel for MRI and PET. To summarize the results of the meta-analysis, we could identify specifically neural correlates, prototypical brain networks which are involved in each of the diseases, and these are dissociated, regionally dissociated, so we can conclude that imaging biomarkers are a valid tool to be incorporated into diagnostic criteria. Okay, let's now go to individual prediction. So far, I have shown you results of group studies. However, in clinical practice, we are interested in single subjects. One example is Friedrich Nietzsche, who was born and is buried close to Leipzig, and he was discussed to suffer from behavioral variant frontotemporal dementia. Predicting patients individually is done in the framework of personalized or tailored medicine. We have different kinds of prediction. So we can predict diseases, we can predict symptoms, we can predict differential diagnosis. We could even predict conversion from pre-stages to dementia or survival time. And we can also predict treatment success or side effects for differential therapy. I want to show you some examples. We used here support vector machine classification, multivariate pattern classification as a tool. Data originated from the FTLD Consortium Germany and the ADNI database. So let's start with disease prediction. These are the results to predict behavioral variant frontotemporal dementia versus healthy controls. In the upper panel, you see atrophy in patients compared to healthy controls. And in the lower panel, you can see brain regions attributing to correct classification. Here we can reach a high classification accuracy of 82% in comparison to a chance level of 50%, which might even be increased if you go from whole brain classification to meta-analytically driven classification, where we focused only on the regions which were identified in the meta-analysis as disease-specific. These are the results comparing semantic variant primary progressive aphasia and healthy controls. You see there is a big atrophy in the temporal lobe, and we have a high classification accuracy of 97% across the whole brain, and in meta-analytical ROIs, 100%. The classification between diseases and controls works well. Here are the results for the other diseases which are discussed in the FTLD spectrum. we can increase accuracy if we go to region of interest, analyses based on meta-analytical regions, and if we add additionally to structural MRI, FDG PET data, so data for metabolism. However, in clinical practice, we are more interested in differential diagnosis. For instance, separating the different primary progressive aphasia subtypes. Here I show you the results for semantic variant PPA in comparison to non-fluent achromatic variant PPA, and we can also reach here a good classification accuracy of 78%. If we go to the non-fluent variants of PPA, this is logopenic variant versus non-fluent achromatic variant. However, we have problems if we use whole brain approaches. There we can reach only 55%. However, we can increase the classification accuracy if you focus on prototypical networks involved in the diseases as identified in the meta-analysis. Moreover, we have developed classifiers including 7 different diseases and a control group. And here we could reach a high accuracy of 44% with a chance level of 12.5%. So this is necessary to translate all these approaches into clinical routine in my mind. On the other hand, you can also identify symptoms and predict symptoms based on imaging data. Here we show this for the so-called anarchic or alien hand syndrome, where people have the feeling that the hand is not belonging to themselves or the hand is doing movements which are unintended by the subject. So we can identify and predict from imaging data whether these patients with corticobasal syndrome suffer from alien or anarchic limb syndrome. On the other hand, we can also predict treatment success. Here I show an example for Parkinson's disease. In the upper panel, you see a comparison between weak and strong responders to levodopa treatment, identifying regions which are atrophic in weak responders. And in the lower panel, in the multivariate analysis, we show the regions which predict whether people with Parkinson's disease predict well to levodopa treatment or not. So atrophy leads to a weaker response to levodopa treatment in Parkinson's disease. Finally, I want to discuss the perspectives in these approaches. I have shown you results from an ideal world scenario. So we can show that imaging markers reliably differentiate frontotemporal lobar degeneration subtypes. We can also show that dementia syndromes can be predicted individually in single subjects, and that we can predict treatment efficacy individually. However, there has been a discussion about methodological limitations of all these multivariate approaches. So in the real world, we are confronted with many different diseases, let's say 20 different neurodegenerative diseases. And as far as classifiers contain only a small number of diseases, these classifiers are stupid. They can only recognize diseases which are contained in the classifier. And this is contrary to each radiologist. Moreover, dementia syndromes are very fuzzy moving objects because in the dynamical course, the diagnosis can even change. Therefore, we need dynamic disease models with big data to classify correctly in clinical routine diseases. A very interesting discussion here is how diseases shall be defined as entities. So what is the most important focus? Clinical features, imaging features, or molecular features? And in my mind, the stance has to depend on perspective. So if we are interested in predicting behavioral therapy or adapting behavioral therapy, we have to focus on clinical and imaging data. Whereas if we are interested in molecularly specifically acting drug treatment, we shall focus on a molecular stance and molecular features. Thanks a lot for your attention, and my cooperators and contributors
Hi everyone, my name is Choong-Wan, and today I'm going to talk about good practices in developing neuroimaging biomarkers. So the use of machine learning in translational neuroimaging has rapidly increased in recent years, showing its increasing popularity. It is mainly because of the power of neuroimaging and machine learning techniques. And first, the functional neuroimaging, especially fMRI, allows us to do systems level brain imaging with a reasonable spatial temporal resolution. And it also provides a unique opportunity for multi-species neuroimaging with which we can translate our research findings from animal studies to human and human studies to animal. And with machine learning, we can now model high-dimensional data such as functional neuroimaging data to predict some useful outcomes of interest. So combining these two powerful methods provides a pathway to clinical translation, serve as a bridge between this preclinical and clinical research. However, not many clinical tools are available yet, and I want to discuss how we can improve this situation. So there could be many different reasons, but my take on this have three main points. The first one is treating neuroimaging maps as research objects. So this plot shows the survey results from a recent review paper. This shows the classification performance of neuroimaging models for different brain disorders. The first look seems not that bad, and there seems to be many good models. However, this is the results at the model development stage. That means the results were with training data. And if we put this within the biomarker development framework, where most promising models should be carried forward until we achieve the population level generalization, we can find most of the models are stuck at the model development stage. There could be many reasons for that. And you might think that the sample size, that is the amount of data, might be the biggest challenge here. However, when we isolate the multi-site studies, which you should have enough data, you can see the sample size is not the main issue here. The proportion, as you can see, the proportion of studies with prospective testing was still low, even for multi-site studies. And the model performance with the prospective testing was lower than the performance at the model's development stage, as you can see here, suggesting there is a clear bias in the studies. Interestingly, when we examine the promising models that already went through the prospective validation and robust replications, as you can see here, many of them had their names in showing these researchers' intention to share this model further with other researchers and other laboratories. So the point is they treated these models as research objects, okay? So when I shared this interesting observation with my colleagues a while ago, one of them taught me about the boundary object, the concept boundary object. And the boundary object is a term from sociology, and it indicates an object that could be used in different ways by different groups, okay? And these objects are plastic, but robust enough to maintain their integrity. And what I propose here is that we need to treat these brain maps as boundary objects that can be used differently by different groups. These different groups, like for example, cognitive neuroscientists can use these brain maps to study the brain mechanisms, while clinicians can use them for diagnosis, for example. So to promote these flexible uses of the brain maps, we need to share our maps and models through these platforms. And recently I came across this tweet that was advertising a meeting, and I liked its title a lot. The title says, "Scientific Publication Beyond the Text, Sharing Research Objects." So by treating brain maps and models as research objects, we will be able to make the science of neuroimaging more than publication. So second point is we need to focus on clinical utility. If we examine what the machine learning models were designed for in these translational neuroimaging research, you can see most of them were designed for a diagnosis. But will these diagnostic models be clinically informative? I don't think so, because there are already many available and cheaper options for diagnosis. Rather, we believe that these 25% of uses, including predicting treatment responses and prognosis and so on, will be more useful and more challenging. So what I want to emphasize here is that the accuracy is just one among the many factors that can influence the machine automation or clinical translation. So in this machine automation map from Harvard Business Review, there is a, so you can see here, there is an additional factor, risk, in addition to accuracy. So for example, you can see credit card fraud detection here that has a very low accuracy, but the risk is also very low. Therefore, it has already been implemented and used in the real world. And however, the driverless cars, they already achieved really high accuracy, right, like this, but the risk is also high. Because of that, it hasn't been automated yet. Similarly, I think clinical utility should be an important factor for clinical translation here, in addition to the accuracy. So for example, the diagnostic test, even though that has a high accuracy, because of its low clinical utility, I think it won't be translated easily. But early detection and risk assessment, these could be translated more easily, even with their low accuracy, because of their high clinical utility. So therefore, we need to concentrate our efforts on this neuroimaging model with high clinical utility, aims for this risk assessment, conversion prediction, and so on. Okay. The last point that I want to make is that we need to make advances in the knowledge of a human brain using these models. So that is related to the issue of interpretability or interpretation of neuroimaging-based machine learning models. And these models, even with linear models, linear methods, are highly complicated. And it's really hard to know what that means and why and how a model works. But if we cannot really answer this question, we cannot know when the model will fail, or to which individuals or subgroups it applies, and how it can advance our understanding of the neurobiological mechanisms. So for a model to be interpretable, it should be A, readable and understandable to humans, to researchers, and B, provide useful information about neural representations and mechanisms of mental constructs and behaviors, and C, that should show that predictions are based on true brain signal, not noise. In our recent paper, we approached this problem with a multi-level model interpretation framework. And in this, we divided the assessment into 3 different levels: feature level, model level, and biology level. And the biology level assessment is most difficult. And if the existing literature cannot explain the model, we should push the boundary of the current knowledge and pursue the new novel discovery and theory development through new experiments. And the first table of this paper, in this first table of the paper, we added detailed explanation of each level assessment with references for example methods, so that you can find the details later. And the last proposal is the component process approach. And the first, the direct prediction approach uses the neuroimaging features to directly model the clinical outcomes. But in the component process approach, we can first model the intermediate component processes that are more closely linked to the brain functions and use them to model more complicated, complex clinical outcomes. So these color scales are an analogy where the direct prediction approach is a grayscale because it is using only one dimension scale, while the component process approach is RGB color scale, where we can use these component process markers to draw more complicated colors or pictures. That is the clinical outcomes. And with this component process approach, we might be able to connect the Death Valley of translational research because these component process markers would satisfy neuroscientists or cognitive neuroscientists because it can be used to study brain mechanisms, and also could satisfy clinicians because it can serve as ingredients to explain more complicated clinical features. So that's all for my take, and there is another recommended reading, a blog post by Peter Bandettini. I read this blog post a while ago, and I read that today again, and it was worth reading, and I recommended this article. And these are the summary recommendations that I had in my abstract, and I already explained them before, and so I won't repeat them again, but these are the nice summary recommendations. Thank you for listening, and thanks to my lab. Thank you so much. And it's 11 p.m. in Korea, so it's quite late, but thanks for listening. Thank you.